Hello, microbiology students, and welcome to Chapter 15's lesson involving microbial mechanisms of pathogenicity. Now, the term pathogenicity is the ability of something to cause a disease, whereas virulence is how effectively or to what degree that thing is pathogenic. There are portals of entry which something can enter the body, such as mucous membranes or through the skin, as well as parenteral route, which is when things are deposited directly into tissues when barriers are penetrated, such as in a stab wound or something falling on someone, ripping open their skin, breaking a bone, etc. Most pathogens have a preferred portal of entry to enter the body. Two terms for measuring invading microbes or relevant values. There is ID50s, which is how much of an infection is needed to typically result in infectious symptoms in a particular population. I should restate that. ID50s are when there is enough, is how much of something is needed to infect about 50% of a test population. And this shows how virulent a particular microbe is at certain concentrations. LD50s are how much of something is needed to kill off 50% of a test population, typically mice, and it measures the potency of a particular toxin. The first measures how much typically results in infection half the time and how much typically results in death half the time. As far as adherence, almost all pathogens attach to host tissues in a process called adherence or adhesion. Adhesins, which are ligands or ligands on the pathogen, bind to receptors on the host cell, such as on the glycocalyx, or the fimbriae. Now, microbes that form biofilms form communities that share nutrients within their host. Capsules, which are glycocalyx around the cell wall. They impair phagocytosis, or the engulfment of bacteria by large white blood cells. It's shown in the case of Streptococcus pneumonia, which causes pneumonia, Haemophilus influenzae, which can cause pneumonia and meningitis, Bacillus anthracis, which can cause anthrax, and Yersinia pestis, which can cause plague. Now, as far as components of cell walls, there are M proteins which resist phagocytosis by white blood cells, such as these are observed in the cell walls of Streptococcus pyogenes. There are OPA, which are proteins that allow the attachment to host cells, such as in the cell walls of Neisseria gonorrhoeae. And then there are waxy lipid molecules, typically mycolic acid, which helps those microbes resist digestion, such as in the case of Mycobacterium tuberculosis. Enzymes involved include coagulases, which coagulate fibrinogen. There are kinases, which help digest fibrin clots, hyaluronidase, which help digest polysaccharides that hold cells together, collagenase, which helps break down collagen, and IgA proteases, which helps destroy immunoglobulin A antibodies. Now, what's being shown here is a chain reaction between proteins and the tissues that they act on, eventually resulting, in this case, fibrin breakdown of a clot. Antigenic variation. This is when pathogens alter their surface antigens and antibodies are rendered ineffective against that particular pathogen. This is something that happens quite commonly with the common cold, where the common cold is very effective at changing the antigens on its surface so that antibodies the body originally produced are no longer effective at registering the presence of that modified common cold virus. Now, penetration into the host cell's cytoskeleton. These can involve evasins, which are surface proteins produced by bacteria that rearrange actin filaments of the cytoskeleton and can cause membrane ruffling, allowing for puncture into the cell. They use actin to move from one cell to the next, such as in Shigella and Listeria. Now, when using host nutrients, there are cytopathic, which are proteins secreted by pathogens that bind to iron more tightly than the host cell itself. This is relevant because iron is a critical necessity for most pathogenic bacteria. This is one example of a bacterial cytopathic that likes to bind to iron in the body and maintain it for the bacteria's use instead of the host. This can result in direct damage, which disrupts host cell function. It uses the host cell's own nutrients and resources, preventing the host cell to have access to them. It also typically produces waste products that are harmful to the host cell or the surrounding cells, and multiplies. And when pathogens multiply in host cells, like in the case of viruses, these can cause ruptures, which kills that actual cell. Another hazard with pathogens is the production of toxins. These are defined as poisonous substances produced by microorganisms, and presence of these toxins can cause many varieties of symptoms, including affecting the heart function, causing diarrhea, causing shock, which causes significant damage and death, or producing things like fever and other symptoms. There is toxogenicity, which is the ability of a microorganism to produce a particular toxin. And the condition of toxemia is when the presence of a toxin makes it into the host's blood. Intoxications are presence of toxins without microbial growth involved. Exotoxins. They are proteins produced and secreted by bacteria, and typically they're soluble in bodily fluids. However, they destroy host cells and inhibit natural metabolic functions. Antitoxins are antibodies against specific exotoxins, and toxoids are essentially inactivated exotoxins that are no longer functional, and they can be used in vaccines to train the body to tell when certain toxoids, or when certain toxins are present, and try to find the cause or remove those toxins. Now, there are AB toxins that contain an enzyme component in A part and a binding component B part, such as in the case of the toxin by diphtheria. Essentially, these work to help spread or move a toxin from one cell to another. There are types of toxins that disrupt the membrane by lysing the host cells, which basically means ripping open the plasma membrane. There are leukocidins, which kill phagocytic leukocytes, or anything that essentially engulfs that toxin. There are hemolysins that kill erythrocytes, blood cells, by forming protein channels that cause the cells to leak out. And then there are streptolysins, where these homolysins that are produced specifically by streptococci. There are things called superantigens that cause an intense immune response due to release of cytokines from the host cells, such as from T cells. These can cause symptoms of fever, nausea, vomiting, and a variety of other things, including death itself. There are genotoxins that cause damage to a host's DNA, which can result in mutations, preventing the typical action of cell division, or even leading to situations such as uncontrolled cell growth, which is cancer. Endotoxins such as lipid A, portion of lipopolysaccharides of gram-negative bacteria. They are released during bacterial multiplication and when gram-negative bacteria die. They stimulate macrophages to release cytokines and the cause disseminated intravascular coagulation, causing blood clots in the blood vessels. Now, other types of endotoxins could be limulus amoebocyte lysate assay, which is a way of measuring endotoxins or to test their presence. For example, blood of a horseshoe crab contains amoebocytes. Those amoebocytes lyse in the presence of a particular endotoxin, producing a clot, which shows the presence of those original amoebocytes. Plasmids may carry genes for toxins, resulting in the production of antibiotics and enzymes. Plasmids are simply circular chunks of genetic material that can be passed down or inserted from one microbe to another, or one organism to another. Lysogenic conversion involves changes in characteristics of a microbe due to the incorporation of a prophage, where some virus inserted its genetic material that eventually altered the genetic material of the host bacteria, causing potential changes such as inserting genes for greater resistance to certain antibiotics, or making the cell stronger or weaker to certain conditions. Now, getting into cytopathic effects of viruses, these are the visible effects of a viral infection on a cell. These involve 9 particular effects. These include stopping cell synthesis, the production of new cells, causing cell lysosomes to release enzymes, creating inclusion bodies in the cell cytoplasm, resulting in the fusion of cells to create a syncytium, changing the host cell's function, or inducing chromosomal changes in the host cell's genetic code, inducing antigenic changes on the cell surface, including the surface proteins on that particular organism, plasma membrane, or cell wall in certain cases, a loss of contact inhibition in the cell, which can eventually lead to cancer, such as when cells, instead of growing in a particular, say, sheath, like to eventually clump up and resulting in maybe tumors. And also producing interferons that protect the uninfected cells. Now, pathogenic properties of other things. In the case of fungi, they can involve toxic metabolic products that, when digested, break down into toxic components. They may provoke an allergic response in the body. In the case of trichothiocin toxins, they inhibit protein synthesis, or the production of large proteins from amino acids. They may have proteases that modify the host cell membrane, adding or modifying the proteins on the surface of that membrane. And they may have capsules that prevent phagocytosis, which hampers the body's ability to digest, or by white blood cells to engulf and remove infectious agents. There are ergot that are alkaloid toxins that cause hallucinations. There are aflatoxin, which is a carcinogenic toxin produced by Aspergillus. There are mycotoxins that are produced by mushrooms and are neurotoxic, affecting the nervous system, such as pallidin and amanitin. When looking at protozoa, microscopic animals, the presence of protozoa and their waste products can cause many symptoms. The hosts, they typically are excellent at avoiding the host's natural immunity and defenses, either by digesting cells and tissue fluids, growing in phagocytes, or through a process of antigenic variation, where they change their outside surface antigens that current host cell antibodies no longer recognize. For helminths, they may use host tissue for growth, which produces, and when they produce large masses, this can cause significant cellular damage. They also produce waste products, and these waste products can cause a variety of symptoms that also further damage the host. Algae can at times result or produce kinds of neurotoxins called saxitoxins, such as what might be experienced from eating ill-prepared shellfish and having some paralytic effect. Lastly, portals of exit. Well, these are ways where these pathogens can either be removed or escape the body after growing. Some examples include the respiratory tract, where we breathe, such as the process of coughing and sneezing to get a pathogen out of our bodies, or through the gastrointestinal tract, through saliva or through feces. the genitourinary tract, which involves urine or secretions from the penis and vagina area. Also could be from secretions from the skin or sweat glands. And also if blood is leaked, such as when arthropods bite or through needles or syringes that are shared between multiple people, or when an arthropod, such as a mosquito, bites one infected person and then goes on to bite someone else. And this concludes this lesson in Chapter 15 concerning microbial mechanisms and pathways for pathogenicity. Wishing you all well in the next lesson and learning module. Peace.